So we have understood the process of transcription, how DNA helps in the formation of an mRNA. Now we go into a beautiful process called as translation, how the mRNA helps in formation of a protein chain, also called as a polypeptide chain. DNA undergoes transcription to form mRNA in the nucleus. The mRNA goes out of the nucleus into the cytoplasm and goes towards the endoplasmic reticulum and fits into a ribosome. In the ribosome, which is made of rRNAs, mRNA and tRNA help in the process of translation or formation of a protein chain to take place. Let us understand how that process takes place. Translation is defined. Translation is defined as a process in which sequence of codons on the mRNA strand are read or decoded, and accordingly the amino acids are joined to each other to form a polypeptide chain that makes a protein. So in translation, sequence of codons on the mRNA are decoded and sequence of codons is converted into sequence of amino acids. So we read each codon and attract an amino acid to form a polypeptide chain or a protein. Translation refers to the process of polymerization of amino acids, forming a chain of amino acids to form a polypeptide chain or a protein chain. What do you mean by that, sir? Sir, सबसे पहले what will happen? There is activation of amino acids and formation of an amino acid tRNA complex. In the presence of an enzyme, amino acid tRNA synthetase, the amino acid number 1 is activated and each amino acid is attached to a specific tRNA at the C3 end to form an amino acid tRNA complex which needs ATP, and that is called charging of tRNA or amino acylation of the tRNA. Now, what do you mean by this, sir? Sir, ये था आपका DNA and DNA underwent transcription to form an mRNA which enters into the cytoplasm. And mRNA enters into the cytoplasm, goes towards endoplasmic reticulum and goes and fits into a ribosome. So mRNA has fitted into a ribosome. सबसे पहले mRNA fits into a ribosomal unit called as the smaller subunit. तो larger subunit अभी तक है ही नहीं. So what is the ribosome having over it? Smaller subunit 30S in case of prokaryotes and 40S in case of eukaryotes. So we are having only the smaller subunit. Smaller subunit has a groove or a cleft, और उस क्लेफ्ट के अंदर जाके mRNA फिक्स हो जाता है and mRNA gets fixed into the cleft. सबसे पहले एक enzyme called as amino acid tRNA synthetase enzyme. ये enzyme याद रखना, amino acid tRNA synthetase enzyme reads the nitrogen sequence, and that nitrogen sequence is read, and समझो it is GGG, and GGG is read by amino acid tRNA synthetase enzyme. Is GGG the start codon? No. And of course, it could be anything. It could be GGG, it could be CCC, it could be CGA, it could be ACG. कुछ भी हो सकता है, but that is not the start codon. So that is called as the UTR. UTR, untranslated region. Untranslated region which is found before the start codon, and as a matter of fact after the stop codon. So UAA stop codon है, उसके बाद भी GGG है, तो it won't be read, and that is also called as the UTR. So start codon AUG के पहले होता है untranslated region, stop codon UAA और UAG और UGA इनके बाद भी होता है a sequence, and that is say GGG or GCG or something which is not read and is called as the UTR, untranslated region. So we have start codon के पहले UTR and stop codon के बाद में also you have UTR. And UTR does not get translated. Then comes AUG. AUG is the start codon. It is read by amino acid tRNA synthetase. And for AUG, the first amino acid in the amino acid pool, methionine, gets activated. Methionine, which is the start amino acid. By the way, that is in eukaryotes. If it is a prokaryote, F-methionine, formilated methionine, gets activated by amino acid tRNA synthetase. This amino acid, methionine, gets activated by ATP. ATP activates it, and this amino acid attaches to a tRNA at its C3 end, to its CCA, to a tRNA having a complementary anticodon. So if the codon is AUG, the anticodon is UAC, and amino acid attached to tRNA with complementary anticodon UAC is now a complex form which is called as an amino acid tRNA complex. An amino acid tRNA complex using ATP, and this tRNA goes and fits towards the codon. So anticodon goes towards the codon, and here you have AUG, and here you have UAC, and the tRNA brings the amino acid methionine. Now, methionine is delivered as soon as one start codon AUG and start anticodon UAC bond, and methionine is delivered, the larger subunit develops with the three sites. जैसे ही first codon और first anticodon attach हो जाते हैं, now develops the larger subunit. तो पहले लार्जर सब यूनिट था ही नहीं. There was no larger subunit. Only after the first amino acid tRNA complex goes and fits into the ribosome, where start codon pairs with start anticodon and the first amino acid is delivered, now develops the larger subunit. तो अब larger subunit develop होता है after the first amino acid is delivered, and that is the process of initiation of translation. Now, as soon as the larger subunit develops, check this here. It has got 1, 2, 3, a UTR, AUG, the first codon, and the second codon. So totally how many nucleotides are there? 1, 2, 3 as the UTR, AUG as the first codon, and first, second, third, three nucleotides as the second codon. So a total of 9 nucleotides representing approximately 3 codons are present in the smaller subunit of the mRNA. The smaller subunit has UTR, first codon, second codon. And in the larger subunit, develop 3 hypothetical parking spaces. 3 hypothetical parking spaces called as the E site or exit site, the P site or peptidyl site, and A site or amino acid site. तो सर स्मॉलर सब यूनिट है ना कॉम्पैक्ट होता है, स्मॉलर सब यूनिट कॉम्पैक्ट होता है, लार्जर सब यूनिट एम्प्टी होता है, लार्जर सब यूनिट खाली होता है. सो दिस इज समथिंग लाइक अ solitaire diamond ring. So you have a solitaire, a diamond, and that is compact, and attached to it is a gold ring, and the gold ring is empty, in which your finger can be put inside. So this is a ribosome. तो smaller subunit compact है, larger subunit खाली है, empty है, and in that solitary diamond ring we make a hole. तो वो डायमंड के अंदर एक होल बना के उसमें एक नीडल पास करते हैं. तो सॉलिटेयर का डायमंड है, नीचे गोल्ड रिंग है, उसके अंदर एक होल बना के नीडल पास करते हैं. व्हाट इज द होल कॉल्ड एज? क्लेफ्ट. द होल इज कॉल्ड एज क्लेफ्ट. एंड व्हाट इज द नीडल which passes in it called as? It is called as the mRNA. mRNA has fitted at which end? It has fitted at C5 end. तो C5 end पे mRNA fit हुआ है, and far away is the C3 end. So mRNA has fitted at which end? C5 end, and very far away is the C3 end, and you have UTR, you have start codon, and you have second codon. So a total of 3 nucleotides के 3 sequences है, and that is UTR, first codon, second codon, which corresponds to sites called as E site, corresponds to site called as P site, and site called as A site. So you have E site, you have P site, and you have A site. Where is amino acid methionine delivered? In the site called as P site. P site पे amino acid methionine deliver हो जाता है. And when the larger subunit attaches to the smaller subunit, it uses GTP as a source of energy for binding the larger subunit with the smaller subunit. उसमें ATP नहीं, उसमें GTP use होता है. And smaller subunit is 30S, smaller subunit is 50S, and it forms a ribosome that is 70S in case of prokaryotes. So ये prokaryotic. Vishal sir had taught you that. 30S smaller subunit, 50S larger subunit, 70S ribosome. If it's a eukaryote, then smaller subunit is 40S, larger subunit is 60S, and the ribosome is 80 Swedberg units, ATS. तो ये तरह से प्रोकैरियोटिक या यूकैरियोटिक राइबोसोम बन जाता है. And now what will happen? So you had first codon read and first amino acid transferred. Now the second codon is read. Second codon can be anything. Let us assume it is GGG. GGG is the second codon. It is read by amino acid tRNA synthetase. Amino acid tRNA synthetase. Message goes by amino acid tRNA synthetase into the amino acid pool. And for GGG, we remember the amino acid activated will be glycine. Glycine gets activated, it gets attached to a tRNA at the C3 end to the CCA having complementary anticodon. And that amino acid tRNA complex now moves into the site called as the A site. तो A site पे वो आ जाता है, codon is GGG, anticodon will be CCC. Between GC triplet, GC triplet, GC triplet, and between UAC क्या हुआ था, if you remember, AU doublet, UA doublet, GC triplet. तो ऐसे कोडॉन एंटीकोडॉन का पेयरिंग होता है, एंड ये है CCC, एंड अमीनो एसिड glycine gets delivered. So, for AUG, which amino acid got delivered? Methionine, which is amino acid number 1. For GGG, which amino acid got transferred? Glycine, which is amino acid 2. Methionine कौन से साइट पे आया है? Methionine has come towards the P site. And new amino acid glycine. Glycine comes at which site? Glycine comes at A site. So new amino acid always comes in the A site. New amino acid always comes in the A site. So we have E site, which is corresponding to the UTR. P site corresponds to methionine, which is the first amino acid. GGG, second codon, has A site corresponding to the second codon with amino acid glycine delivered as the second amino acid. And now there is an enzyme called as peptide transferase enzyme. Peptide transferase enzyme transfers amino acid number 1, methionine, from the P site to the A site. तो यहां था methionine, and methionine gets delivered from the P site to the A site. तो A site पे we already had glycine, अब जाके उसके पास methionine भी आ जाता है, और ग्लाइसिन और मिथियोनिन के बीच में बन जाता है पेप्टाइड बॉन्ड. तो A site, which has the second codon, and P site, which was corresponding to the first codon. Sir, अब क्या होता है? Amino acid methionine gets delivered to glycine, और glycine और methionine के बीच में बन जाता है पेप्टाइड बॉन्ड. Which enzyme did the delivery? The enzyme which did the delivery was peptide transferase enzyme. Peptide transferase enzyme transfers amino acid methionine from the P site to the A site. From P site to A site, the delivery was done by peptide transferase enzyme, and glycine amino acid 2 and methionine amino acid 1 के बीच में बन जाता है peptide bond. So at the moment, E site is corresponding to UTR. It is empty. The P site has tRNA with no amino acid corresponding to first codon AUG, and second codon GGG has A site, and A site has got two amino acids, A site has got 2 amino acids. So E site is empty, P site has only tRNA, A site has tRNA with 2 amino acids. And now takes place translocation. The ribosome moves from the C5 end to the C3 end by 1 codon. तो राइबोसोम यहां था and ribosome shifts by 1 codon. Ribosome shifts by 1 codon. So, what was the ribosome initially occupying? UTR, AUG, GGG. This was present first in the ribosome. ये राइबोसोम में पहले था. Now, as the ribosome has shifted by 1 codon, what will the ribosome occupy? ये है ribosome, ये है mRNA, and from the C5 end to the C3 end, ribosome shifts by 1 codon. ये देखना ध्यान से. Ribosome shifts by 1 codon from the C5 end to the C3 end. Ribosome shifts by 1 codon, and that phenomenon is called as translocation. That is called translocation. तो ध्यान से ट्रांसलोकेशन पे क्या होगा? सर ट्रांसलोकेशन पे राइबोसोम शिफ्ट्स बाय 1 codon. So initially it was UTR first codon and second codon. Now, as ribosome shifts, it will occupy first codon AUG, second codon GGG, and say third codon, say it is UUU. So it occupies first codon, second codon, and third codon. So instead of occupying UTR first, second, it is now occupying first, second, third. First, second, third. So UTR 1, 2 की जगह it occupies 1, 2, 3. And that is called as translocation. What will happen in the larger subunit? Sir, initially this was P site, sorry, E site, this was P site, and this was A site. तो UTR के corresponding था E site and AUG first codon के साथ था P site and GGG second codon के साथ था A site. Now, as ribosome has shifted, the E site is the old E site, and the P site becomes old P site becomes the new E site. Old P site becomes the new E site. This is the old E site. But अब जब राइबोसोम शिफ्ट हो गया है, the old P site becomes the new E site. The old A site becomes the new P site. And there is a new A site. So once again, sir, you have UTR first, second. And as the ribosome has shifted, you have first, second, third. तो पहले ये क्या था? E site, P site, A site. As ribosome shifts by 1 codon, the old E site goes out of the ribosome and the P site becomes the new E site. The A site becomes the new P site. And there is a new A site. So once again, this was old E site, old P site, old A site, and this was the UTR first second. As the ribosome has shifted, the old E site goes out of the ribosome. The old P site becomes the new E site. The old A site becomes the new P site. And there is a new A site. The E site corresponds to first codon. E site corresponds to first codon. The P site corresponds to the second codon. And the A site corresponds to the third codon. So E site corresponds to first codon. A site corresponds to P site corresponds to second codon, and A site corresponds to the third codon. So what do we have at the P site? At the P site, you have only tRNA. First codon, first anticodon, only tRNA. At the E site, at the P site, we have tRNA with two amino acids, glycine and methionine. If you remember, वो तो यहाँ पर ही हो गया था before translocation, that peptide transferase had transferred amino acid, and we have two amino acids, and this tRNA has lost its amino acid. So now on translocation, what will happen? At the E site, we have tRNA with no amino acid. At P site, we have tRNA with two amino acids. And A site corresponds to a third codon. A site corresponds to third codon. So first codon corresponds to the old first codon corresponds to the E site. First codon corresponds to E site. Second codon corresponds to the P site. And third codon corresponds to the A site. And now what will happen? Sir, AUG, first codon, and second codon जो था वो तो भी empty हो गया है, तो उसका कुछ काम नहीं है. That tRNA exits the ribosome from the E site. E site से ribosome will release the tRNA. So tRNA exits from the E site, and that is the reason E site is called as the exit site. tRNA exits from the E site, and that is the reason E site is called as the exit site. And the second codon is read. Second codon UUU. Message goes in the amino acid pool, and in the amino acid, an amino acid gets activated. If you remember which enzyme? Amino acid tRNA synthetase enzyme. कौन सा enzyme? Amino acid tRNA synthetase enzyme. कौन सा enzyme हमेशा act करता है? Amino acid tRNA synthetase enzyme. And amino acid tRNA synthetase enzyme will read UUU. Message goes into the amino acid pool, and for UUU, the amino acid activated is phenylalanine, the third amino acid. It will get attached to a tRNA at its C3 end and enter into the ribosome. The new tRNA always enters at the A site. tRNA enters at the A site. So UUU, AAA, UA double bond, UA double bond, UA double bond, codon pairs with anticodon, and the tRNA delivers new amino acid phenylalanine at the A site. Golden words. Where does tRNA exit from? tRNA exits from the E site. tRNA exits from the E site. So it is called exit site. Where does tRNA enter? tRNA enters from the A site. tRNA enters in the A site. And what is the P site going to do now? The two tRNAs from the P site go and get attached to the tRNA at the A site. And that is done by peptide transferase enzyme. Peptide transferase will transfer amino acid from P site to the A site. So that is the reason it is called P site peptidal site. पेप्टिडाइल साइट पे पेप्टिडाइल ट्रांसफरेज एक्ट करता है, सो इट इज कॉल्ड पेप्टिडाइल साइट. और अमाइनो एसिड का पॉलीमराइजेशन होता है, अमाइनो एसिड सीक्वेंस का चेन बनता है, एंड दैट इज कॉल्ड अमाइनो एसिलेशन. amino acylation. So amino acylation or formation of protein chain or polypeptide chain takes place at which site? It takes place at A site. So once again, where does tRNA exit from? tRNA exits from E site. So it is called exit site. Where does tRNA get transferred by peptidal transferase from? tRNA gets transferred by peptidal transferase from P site. So peptidal transferase acts at which site? P site. So it is called as peptidal site. And where does amino acid gets transferred to? To the A site. So, where does amino acylation take place? Amino acylation takes place at the A site. Amino acylation takes place at the A site. So, it is rightly called as the amino acyl site. Amino acyl site. And that is the way the protein synthesis takes place. So, translocation takes place again. So now ribosome shifts by one codon, and codons, initially the ribosome was occupying first codon, second codon, third codon. Now as translocation takes place, it will occupy second codon, third codon, fourth codon. एंड ये है आपका ई साइट, ये है आपका पी साइट, ये है आपका ए साइट. तो tRNA exits from the E site. tRNA exits from the E site. A fourth codon is read, CCC. And for CCC, new amino acid gets activated, say proline. And proline is delivered by tRNA having anticodon GGG and CG triplet, CG triplet, CG triplet. And fourth amino acid enters into the A site. And three tRNAs from the P site will now get delivered or transferred to the A site. If you remember, जब translocation हुआ था, on translocation, what will be this site? On translocation, what will be this site? This is P site. What will P site become? New E site. P site will become new E site. What will A site become? New P site. and CCC corresponds to which new site? A site. तो ई साइट पे क्या आ गया? सेकंड कोडॉन. ई साइट पे कौन आ गया? सेकंड कोडॉन. पी साइट पे कौन आ गया? थर्ड कोडॉन. ए साइट पे कौन आ गया? फोर्थ कोडॉन. तो यू हैव सेकंड कोडॉन. ये है यूटीआर. एयूजी फर्स्ट कोडॉन, जीजीजी सेकंड कोडॉन, यूयूयू थर्ड कोडॉन, सीसीसी फोर्थ कोडॉन. फर्स्ट, सेकंड, थर्ड, फोर्थ. तो on translocation, instead of first, second, third, you now have second, third, fourth. And what does tRNA exit from? P site. What does E site? I'm sorry, tRNA exits from E site. What does P site now have? P site has got tRNA with 3 amino acids, and A site got fourth amino acid. And where does peptidyl transferase transfer it? Peptidyl transferase transfers it from P site to the A site. तो इफ यू हैव अंडरस्टूड, हमेशा टीआरएनए कहां से एग्जिट होता है? ई साइट. वेयर डज न्यू टीआरएनए विथ न्यू अमाइनो एसिड ऑलवेज एंटर? ए साइट. ए साइट. And where does the old chain of amino acid transfer to the new chain to form a new chain? From P site to the A site. And which enzyme will do that? Peptide transferase enzyme. So which enzyme transfers amino acid from P site to A site? Enzyme peptide transferase transfers amino acid from P site to A site. तो ये हो गया 3 amino acids, and first, second, third, 3 amino acids on translocation, second, third, fourth. So these are fourth amino acid. तो ये 4 amino acid. इस तरह से ट्रांसलोकेशन होते रहता है. Exit will always take place from E site. So when this moves, समझो it occupies now second codon, third codon, and third codon, fourth codon, and fifth codon. What will this site become? The new E site. What will this site become? The new P site. What will this site become? The new A site. Where will tRNA exit from? E site. Where will new amino acid enter? The fifth amino acid enter into the A site. And where will 4 amino acids go from? 4 amino acids will go from P site to the A site. And that is the way translocation hota jayega. And this process will take place till all the amino acids are read, and on translocation you have a codon called UAA, or you have UAG, or you have UGA coming into the A site. तो ये कोडॉन्स ए साइट पे आता है से UAA, and message is read by amino acid tRNA synthetase. Amino acid tRNA synthetase will read UAA, but in the amino acid pool there is no amino acid which gets activated by UAA or UAG or UGA. No amino acid gets activated, so transfer of amino acid will not take place. The amino acid transfer will not take place, and there will be no transfer from P site to the A site. So transfer nahi hoga. And as a matter of fact, if the A site gets attached a chemical called as release factors. एक रिलीज फैक्टर नाम का केमिकल आता है R1, R2 एंड S. R1, R2 and S release factors come and get attached to the A site. तो A साइट पे रिलीज फैक्टर R1, R2, S आ जाता है and they release the amino acid chain from the P site. तो P साइट से अमाइनो एसिड चेन रिलीज हो जाती है and that is now the amino acid chain released from the P site. So which was the first amino acid? Methionine. Which is second amino acid? Glycine. Which is the third amino acid? Phenylalanine. Say fourth amino acid? Serine. Sixth amino acid? Aspartame. Seventh amino acid? Histidine. Eighth amino acid? Tryptophan. Ninth amino acid? Serine. 10th amino acid? Again serine. 11th amino acid? Again methionine. ऐसे 1000 अमाइनो एसिड का एक चेन बन जाता है एंड सपोज दिस इज द बीटा सेल ऑफ islets of Langerhans of pancreas. What is this new amino acid chain called as? Insulin. If this was the beta cell, then insulin was formed. If the sequence was AUG, GGG, CCC, UUU, CCC, etc. If you had AUG and you had CGA and you had UAA, the UA, you had CUA, तो उससे बनता था मिथियोनिन, टायरोसिन, फेनिलएलनाइन, ग्लाइसिन, एक्सेट्रा एक्सेट्रा, एंड इससे बन जाता था अमाइनो एसिड चेन इंसुलिन की जगह एक्टिन मायोसिन, जिससे मसल्स बनता था. So we had done that before. This is the way amino acid formation sequence takes place. Different proteins are formed. Once an amino acid chain is formed, the tRNA will exit from the P site. tRNA P site में से ही exit हो जाता है and the larger subunit disappears. Larger subunit गिर जाता है and the smaller subunit के अंदर mRNA breaks apart. So mRNA is destroyed immediately after translation has taken place. So the smaller subunit has a cleft again. Larger subunit चला गया है, tRNA चला गया है, mRNA destroy हो चुका है. In the smaller subunit will now fit a new mRNA and a new process of translation will begin. So which RNA has the shortest lifespan? mRNA has the shortest lifespan. It gets immediately destroyed after a translation process has taken place. So mRNA has the shortest lifespan. So, sir, we have understood this beautiful process of translation. And, sir, then we have polysomes or polyribosomes. In order to increase cellular efficiency, many ribosomes are attached to an mRNA to form polypeptide chain, which are called as polyribosome. If you remember, in prokaryotes, so you have a long mRNA which has got start codon, stop codon, start codon, stop codon, start codon, stop codon, and that is called as polycistronic mRNA. Polycistronic mRNA. We have just done that. So it will fit into a ribosome, into another ribosome, into another ribosome. एक राइबोसोम में एक ट्रांसलेशन होगा, दूसरे राइबोसोम में दूसरा ट्रांसलेशन होगा, तीसरे राइबोसोम में तीसरा ट्रांसलेशन होगा, एंड ऐसे एक चेन ऑफ राइबोसोम्स होता है, और चेन ऑफ राइबोसोम में वन सिंगल एमआरएनए अंडरगोस ट्रांसलेशन। तो एक ही सिंगल एमआरएनए का मल्टीपल ट्रांसलेशंस हो रहा है, बिकॉज इट इज अ पॉलीसिस्ट्रॉनिक एमआरएनए, एंड दिस कलेक्शन of multiple ribosome is called as polysome or polyribosome. That is called as polysome or polyribosome. Ribosomes are composed of 65% rRNA and 35% protein. If you remember, a ribosome is made up of rRNA and that is 65%, और उनको स्टिक करने के लिए प्रोटीन्स होते हैं which are 35%, and 35% are there. And ribosome consists of structural RNAs called rRNAs and 80 different types of proteins. How many types of proteins are there? 80 different types of protein. The ribosome also acts as a catalyst. 23S rRNA is the enzyme called ribozyme for the formation of a polypeptide chain. Sir, if you remember, which enzyme is this? 23S rRNA. It will help in formation of peptide bond. So if you remember, in eukaryotes, peptide transferase transfers amino acid from the P site to the A site. That is done by peptide transferase. Peptide transferase forms the peptide bond. In prokaryotes, not peptide transferase, but 23S rRNA. 23S rRNA, which is called as ribozyme. Ribozyme. So याद रखना, rRNAs also act like enzymes. And rRNA not only forms ribosome, it also acts like enzyme. 23S rRNA ribozyme transfers amino acid from the P site to the A site. So ये होता है यूकैरियोट्स में peptidal transferase, and prokaryotes में 23S rRNA is the enzyme ribozyme for the formation of a peptide bond. And the mRNA has additional sequences that are not translated and are referred to as UTR, untranslated region, that we had talked about. UTR are present at 5' end before the start codon and at the C3 end after the stop codon. So ये है आपका mRNA, ये है AUG, then we have codon, 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 and sir, you have UAA as the stop codon. तो start codon, stop codon, start codon के पहले होते हैं UTR, untranslated region, जो ट्रांसलेट नहीं होगा, एंड UAA के बाद में भी होता है UTR. So at the C5 end, before start codon is the UTR, and at the C3 end, after stop codon is the UTR. So you have untranslated region before start codon and after stop codon. Chalo, so we have analyzed beautifully the process of translation. Very nice logic we have understood here. So with that, we have understood the concept of protein synthesis.